Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the recorded sessions, live sessions, study materials, and also concept videos. Moreover, you're going to get this lecture notes in the form of PDFs. Get your acquaintance with all the channels that are made available for you and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, click on the like as well as share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Saying that, let's begin our today's session. We are in the series of the sessions where we have been discussing about the chapter, that is molecular basis of inheritance, which comes into the unit genetics and evolution, right? Now, so in last session, we discussed about the transcription unit, which has different regions like promoter, structural gene and terminator. Promoter is a sequence where the enzyme RNA polymerases is going to bind. And after that, it is followed by the structural gene and ends up with the terminator, which terminate the process of transcription. If you look at the sequence of central dogma, where it starts up with the DNA and DNA can replicate, produce its own copy by means of replication. Now, whatever the genetic information which has been stored in the DNA is now copied into mRNA. And that process is nothing but transcription presently we are dealing with. Now, the genetic information which has been transmitted into mRNA is now going to be used to synthesize the sequence of the amino acids that are present in the structure of protein. That means protein synthesis. And this process is called as translation. So, four major processes of molecular biology of central dogma. Replication, we already studied in detail about the replication of DNA. Now, transcription, in this process, we have understood about the transcription unit. Now, so here, types of RNA and the process of transcription. So, in bacteria, in the last session itself, we mentioned about the differences between the process of transcription that is going to be happening in the prokaryotic cells as well as the process of the transcription that is going to be happening in the eukaryotic cells. The complexity of the process of transcription is in turn with the eukaryotic cell when compared to the prokaryotic cells, because the organization is very simpler in prokaryotic cell when it is compared to the eukaryotic cell. The organization and the presence of the true nucleus can be seen in the eukaryotic cell. So therefore, there is a division of compartments where the process of replication happens and where the process of transcription happens. That can be seen in the case of eukaryotic cells. Let's start up with a simple process of transcription that is happening in the prokaryotic cell. Example for the prokaryotic cell is bacteria. In bacteria, there are three major types of RNAs. We know that mRNA is the RNA which we call it as a messenger RNA that is going to be act as a template for the formation of the proteins. Along with the mRNA, other RNAs are also useful for the production of proteins. Messenger RNA, in short mRNA, which is used as a template. tRNA, transfer RNA, which brings the amino acids that has to be sit and form the sequence of the amino acids, and finally it forms the structure of the protein. Now, rRNA, ribosomal RNA. So, altogether, three different types of RNAs are useful in the process of transcription and while the formation of the proteins are taking place, all this has to be produced and all this has to be ready for the process of protein synthesis, that is called as translation. So here all three RNAs are needed for the synthesis of protein. Synthesis of protein means translation, synthesis of protein, along with the mRNA, which is produced as a result of transcription, which carries all the genetic information that has been stored by DNA, right? So the major source where the storage of the genetic material, which stores the genetic material, that is nucleic acid and that is DNA, which transmits that DNA, that is mRNA. mRNA will do that transmission by copying the stored form of genetic information which is present in the DNA. Now, the genetic information has been carried, that means has been copied and transcribed by the means of messenger RNA. We are telling that along with the messenger RNA, we need tRNA as well as ribosomal RNA, rRNA for the process of synthesis of proteins, that is protein synthesis. Now, here we said that three different types of RNAs we need. For what they are useful? The mRNA, that is messenger RNA, provides you template. We know that DNA, not the entire length of the DNA, only a single segment of the DNA, small segment of the DNA that is going to be transcribed. In that also, only one strand out of the two strands. Only one strand out of the two strands that is acting as a template and it has been transcribed into mRNA. And we are talking about this mRNA. This mRNA is going to act as a template. Now, tRNA, which brings amino acids. We know that amino acids are the building blocks of protein. For the protein to synthesize, it needs the amino acids that has to be sequenced. And to make the sequence of the amino acids, individual amino acids bring to the protein structure to form the sequence of the amino acids, and that is happened by the means of tRNA. So tRNA brings amino acids and also it reads genetic code. Genetic code by which the sequence of the amino acid is decided. And we know that if each single amino acid is changed, it is going to show its impact on the structure of the protein itself. That means the codon will have a capacity to decide which amino acids that has to be placed in order to form the sequence of the amino acids, and that sequence of the amino acids decides the structure of the protein in turn. Now, and finally, ribosomal RNA, which plays a structural as well as functional, and that function is acting as a catalytic role during the translation. And translation is nothing but protein synthesis. Now, there is single DNA dependent RNA polymerases. There is a single DNA dependent RNA polymerases that catalyzes the transcription of all the RNAs. All the RNAs means all three RNAs like tRNA, mRNA and rRNA. Right, in the bacteria. So in bacteria, just now we said that these are the lower organization and prokaryotic cell where we do find only one enzyme that is going to synthesize all the three RNAs, that is tRNA, mRNA and rRNA. Whereas when come to the case of eukaryotic cells, we have three different types of enzymes and also division of labor exists. Which enzyme will synthesize what tRNA? That division can be seen in case of eukaryotes, but in the case of prokaryotes, all the RNAs are synthesized by a single DNA dependent RNA polymerases enzyme. Now, looking here. So, we know that transcription unit consists of three regions, which starts with the promoter, structural gene and terminator, right? RNA polymerases binds to the promoter region, binds to promoter region. And after binding, we already know that the promoter will initiate the process of transcription. Therefore, once the RNA polymerases binds to the promoter and it initiates the process of transcription, that is the first step of transcription, what we call it as a initiation, followed by elongation, followed by termination. So what are the steps that are involved in transcription? Initiation of the transcription, elongation of the chain, and finally termination of the transcription. Now, for this, the substrate, as we already know, while we are discussing about the replication of the DNA itself, we said that nucleoside triphosphates, these are going to act as a substrates. Depending upon the nucleosides which are present on the DNA, taking that DNA as a template, now the nucleoside triphosphates come to form the pairing by means of hydrogen bonds, and these all happens depending upon the complementary base pairing, complementarity. So here it helps nucleoside triphosphate, and it helps the nucleotide triphosphate on what basis it has to come and join. For this, the governing factor is the complementarity. Now, it uses the nucleoside triphosphate, which uses enzyme uses promoter. To the promoter region, enzyme got attached. Now, it uses nucleoside triphosphates as a substrate and polymerizes. Polymerization happens in the direction of 5' to 3', same to the direction of the replication. Now, in a template dependent fashion. Here, depending upon the sequence that is present in the DNA, mRNA is constructed. So, as the formation, polymerization of the RNA is depending upon the sequence of the DNA, we call it as a template. This DNA, one strand of the DNA is acting as a template, right? So, it is a template dependent fashion, and following the rule of complementarity. We already know complementarity is the base pairing. Purine base pairs with the pyrimidines and pyrimidines base pairs with the purines, with a single exception where in the place of thymine that is present in the DNA, here the uracil is coming in the RNA. That is what the rule of complementarity will govern the process of polymerization of the nucleoside triphosphate that has to come on the mRNA, and for this the template is DNA. That is what this process is considered as a template dependent process. Now, so here it somehow also facilitates opening of the helix. DNA is present in helical structure. We need the opening. This opening we need for the process of transcription to proceed. So it somehow also facilitate, which enzyme, RNA polymerases somehow facilitate opening of the helix. Here is the opening of the helix and continue elongation. That is addition of, that is a polymerization reaction continues, that is the elongation of the chain. Elongation of chain means elongation of mRNA. Now, only a short stretch of RNA remain bound to the enzyme. And this is how, once the mRNA is synthesized, that newly formed mRNA will be get detached from the DNA, and now it is going to act as a template for the further process. What is the further process? That is protein synthesis, that is translation. Now, so here, once polymerization reaches the terminator, so it starts with the promoter, continues with the structural gene that is called as elongation of the chain, and finally it reaches to the terminator. Once it reaches to the terminator, which is the end of the process, we call it as the nascent newly formed RNA, that is mRNA, falls off. It separates. See, here is an initiation, and once the process get initiated, the substrates keeps on adding, that is nucleoside triphosphates. Once it is done, the increase in the chain, that is elongation of the chain happens. Once the RNA is formed, the nascent, that is newly formed mRNA falls off. It comes out and this will be our mRNA. Right? Once the nascent RNA falls off, so also the RNA polymerases. RNA polymerase enzymes will also get detached and it will be useful for the another cycle of the transcription. Now, this results in the termination, that it reaches the final process of the transcription and it reaches to the termination of the transcription. That is what we said, that in the transcription we have initiation, elongation and finally termination. Now, so here we can say that this is how RNA polymerases. RNA polymerases is how it is going to accelerate, initiate, initiation, elongation as well as termination. For this, is able to catalyze 3 steps. We have 3 different steps, right? It has to initiate, it has to elongate and it has to terminate. For this, question is, how a single RNA polymerases in the prokaryotic cells, that is bacteria, it is able to do all the steps that are involved in the transcription, that is initiation, elongation and termination. Here is the answer for that. The RNA polymerases is only capable of catalyzing the process of elongation. Then what about initiation as well as termination? All the 3 steps has to be takes place in such a way that it leads to the complete copying of the genetic information from DNA to RNA, and that is what we call it as a mRNA. But here in this transcription process, we have 3 steps: initiation, elongation and termination. This intriguing question where how the RNA polymerases, single RNA polymerases is going to look up all steps that are involved in the process of transcription. It is known that RNA polymerases is capable of catalyzing only single step, that is elongation, that is polymerization, that happens in the direction of 5' to 3'. Then what about initiation as well as termination? Here, it will get associate temporarily, that is transiently, with the factors. 2 important factors. One factor for initiation, that what we call it as an initiation factor, or also called as sigma factor, and other one is termination factor, also called as rho factor. So, it associates transiently with initiation factor as well as termination factor, which associates RNA polymerization associates. It's an enzyme. First associates with a sigma factor. Sigma factor is your initiation factor. Then it initiates first step of the process of transcription. Then it can by itself catalyzes the process, that is the second step, that is elongation of the transcription. Once it reaches towards the end of the transcription, then again it associates. By that time, this sigma factor will be removed off. Now newly it get associates with another factor, and that factor is called as termination factor, which is otherwise called as rho factor. Thereby, by getting associated with initiation factor, it can initiate the process of transcription. By associating with rho factor, otherwise called as termination factor, it can terminate the process of transcription. This is how a single RNA polymerases is going to catalyze all the steps of initiation, elongation of the chain and termination of the transcription in prokaryotics, that is bacterial cells. Association with these factors alters the specificity of the RNA polymerases to either, in that way, it can either initiate the process or terminate the process Now, in bacteria, since mRNA doesn't require any processing, while we are discussing about the splicing, if you look at the higher organization, say for example any eukaryotic cell, there once the mRNA is produced, See, from DNA to the process of, through the process of transcription, there is a copying down of the genetic material which is present in the DNA into mRNA. Whatever the mRNA that is produced in the prokaryotic cell is active. It doesn't require any further processing. But the mRNA which is produced in the eukaryotic cell is initially, it is inactive because it is containing a non-coding sequences, what we call it as an introns. Therefore, joining of the exons and removing of the introns in a definite order, it is very much required in the case of eukaryotic mRNA. That is what we call it as an, initially it is a hnRNA, then it is processed to the fully mature mRNA. But that type of maturing, that is processing of mRNA, is not required in the case of bacteria. In bacteria, since the mRNA does not require any processing, it doesn't require any processing to become active, because the whatever the mRNA that is produced in the prokaryotic is itself in an active form. And also, since transcription and translation takes place in a same compartment. See, if you look at the cell of the bacteria, we don't have a much developed nucleus inside it. Here, cytoplasm is the site where the transcription as well as translation takes place. There is no different compartments for transcription to be happen and translation to be happen. If we come to the case of the eukaryotic cell, where we have a very well developed nucleus there. So there, inside the nucleus, the process of the transcription takes place. And that processed mRNA, it comes out of the nucleus through the nucleopores to reach to the cytoplasm. And the cytoplasm is the site where the protein synthesis, that is translation, happens. There, we find different compartments for the different processes to take place, that is transcription and translation. But when compared to the bacteria, prokaryotic cell, both transcription as well as translation is going to be happen in a one single compartment, because we don't have a well-defined nucleus over here. There is no separation. This is the point to be noted. There is no separation of cytosol, that is cytoplasm, and nucleus in bacteria. So, as a result, it happens in the same compartment. Transcription as well as translation happens in the same compartment. Many times, translation can begin much before the mRNA is fully transcribed. Even before the mRNA is fully transcribed, the transcription has begun and it has not reached the end point. Even before the translation happens. That means it is of coupled processes. Subsequently, so we can say that consequently the transcription and the translation can be coupled in bacteria. So, in eukaryotic cells, we see that the transcription has to be begin and it has to continue and it has to end up properly, which leads to the formation of a mRNA. Just now we said that the mRNA what is produced in the eukaryotic cells are inactive initially. Then again it has to go for the processing. Once the processing done, then only we'll get the active mRNA, and this produced active mRNA comes out of the nucleus and reaches to the cytoplasm. There when there is a beginning of translation. But these are all the cases which will not seen in the case of bacteria. Therefore, even much before the transcription has ended off, translation has already begun. So that is what the transcription and translation can be coupled in bacteria. Now, so here is a diagram where we can see 3 steps which are involved. Initiation, elongation, termination. So, here is a promoter region and the polymerization always happens from the direction of 5' to 3' direction. So, this is DNA which is acting as a template. The red strand is going to act as a template. So, here is a region of promoter to which RNA polymerizes enzyme. And we said that this enzyme can catalyze the elongation. In order to initiate the process, in order to show the step of initiation, RNA polymerase enzyme has to temporarily associate with a factor, and that is called as sigma factor, otherwise initiation factor. Once it is associated with the initiation factor, now promoter to which the RNA polymerase is attached, to which this RNA polymerase is in turn attached with a sigma factor, and it will initiate the process. Now the elongation happens where we can see this is the newly forming mRNA, still forming. We can see the polarization is still happening where it is using the substrate, and the rule here that is called as complementarity. Now, once the elongation is done, now another factor that is called as rho factor or termination factor will get attached to the enzyme, and that is called as RNA polymerases, which leads to termination and releasing of nascent RNA, and that is called as mRNA because it is transferring, it is using as a messenger. That is what we call it as a messenger RNA. So here, so by now we understand one thing is very, very clear, that the process of the transcription is simple comparatively in the prokaryotic cell, but when compared to the coming to the eukaryotic cell, that complexity increases. And what are the reasons for this complexity? Here in eukaryotes, there are two additional complexities. Let's understand what are the complexities that do we find in relation with the eukaryotic cells. There are at least 3 polymerases enzyme. We have only one RNA polymerase that is going to initiate all the initiation, elongation and termination in the case of prokaryotes. But here there is a division of labor in the nucleus, and in addition to the RNA polymerases also we do find in the other cell organelles. So here these 3 RNA polymerases will show the division of labor. There is a clear-cut division of labor, and this is important here. RNA polymerases 1, it is going to transcribe rRNA, that is ribosomal RNA of subunits having, these are important, 28 subunit rRNA, 18 subunit rRNA and 5.8 subunit rRNA. This is RNA polymerase 1. Out of 3, RNA polymerase 1 is going to transcribe rRNA's subunits. Then the rRNA, another RNA polymerases, that is RNA polymerases 2, it is going to transcribe precursor of mRNA. Precursor of mRNA, that is whatever DNA, it is going to transcribe into mRNA. Whatever the RNA, that is mRNA, it is going to be produced initially in the case of eukaryotes are need to be processed, and that we call it as a precursor of mRNA, and that precursor of mRNA is called as heterogeneous nuclear RNA. In short, we call it as an hnRNA. That means, see, DNA will be transcribed into first hnRNA, and this process is catalyzed by RNA polymerases 2. And this hnRNA is a precursor of mRNA. From this, it further undergo processing to produce a mature mRNA, and that mature mRNA is called as mRNA, processed RNA. That is a form which comes before mRNA, that is hnRNA. And this hnRNA is produced, transcribed by means of RNA polymerases 2. So out of 3, first one, it is used to transcribe rRNAs of having subunits 28, 18, 5.8. And second polymerases, that is RNA polymerases 2, is going to transcribe, produce hnRNA, which is a precursor of mRNA. Now, moving on to RNA polymerases 3. RNA polymerases 3 is responsible for transcription of tRNA, 5 subunit rRNA, snRNA. snRNA is small nuclear RNA. So with this, we have different subunits of the rRNAs, tRNA and mRNA. So there is a clear-cut division of labor that can be found in the case of eukaryotic cell, but in the prokaryotic cell, all the steps are catalyzed by a single enzyme, and that is called as DNA-dependent RNA polymerases. Now, moving to the second complexity. First complexity is with relation to the enzyme. We have three different types of RNA polymerases. RNA polymerases 1, 2, 3. Now, the second complexity is that the primary transcript. Primary transcript is hnRNA. Contains both exons and introns, and these introns are non-functional, non-coding. So, think that this is the DNA which transcribes into hnRNA. That is primary transcript. Here in the primary transcript, we can see both exons as well as introns. If you look at here, these are just a minute. Exons, exon and exon. And this part, it is intron, okay? And it is also intron. See, in the primary transcript, we have both exons as well as introns. It has to undergo a process of splicing, where cutting down of the introns takes place and joining of the exons takes place. So here, hence, it is subjected to the process of splicing. Splicing is all about where the introns are removed. Introns are removed. This part removed, okay? Now, we left with exons. And these exons has to be joined together, right? Introns are removed and exons are joined in a definite order. Now, this hnRNA is nothing but primary transcript. hnRNA undergo additional processing. Along with the splicing, it will undergo an additional processing to make it into processed mRNA, and that is capping as well as tailing. Let's see what is capping and what is tailing. We need, see, the point here is whatever the hnRNA, it is a primary transcript and it has to undergo the process of processing. This processing will include splicing, capping, tailing, where hnRNA, heterogeneous nuclear RNA, is going to convert into messenger RNA, that is a processed mature messenger RNA. Now, what do you mean by capping? Capping, it's an unusual nucleotide. Which is that nucleotide? It's an important methyl guanosine triphosphate. Methyl guanosine triphosphate is going to add to the 5' end. Capping is towards the 5' end, tailing is towards the 3' end. This is very important. The tailing, where adenylate residues of length approximately 200 to 300, that is polyadenylate. So here, this adenylate residues are going to add up to the 3' end. This is 3' and this is 5'. So, capping towards 5', tailing towards 3'. In capping, there is an addition of methyl guanosine triphosphate. In tailing, there is an addition of polyadenylates. In a template independent, there is no template for the addition of the polyadenylates. It happens in a template independent manner. Now, let's see. Once the splicing, what do you mean by splicing? Removal of the introns and joining of the exons in a different order, and capping as well as tailing. It is fully processed hnRNA. Now we call it as an mRNA. Once this hnRNA is subjected to splicing, capping, tailing, now we consider that as a fully processed hnRNA, and that is nothing but mRNA that is transported out of the nucleus for translation. In the eukaryotic cell, we have the nucleus, well-defined nucleus. Inside nucleus, this is all happens, where transcription happens, initiation, elongation, termination, splicing, capping, tailing. Once all this happens, now the mature, processed mRNA, which is present inside nucleus, now travels to reach to the cytoplasm. Cytoplasm is a site where translation, protein synthesis happens. Now, the significance of such complexities where just now we discussed the whatever the mRNA which is produced is of primary transcript. It is of hnRNA. It has to undergo the processing which includes like splicing, capping, tailing, and moreover only one enzyme is going to catalyze the process of initiation, elongation, and termination. But here we have three differences, and there is a clear-cut division of labor that can be seen in the case of eukaryotic cell. So this all adds up to the complexity of the process of transcription that do we find in the case of eukaryotic cells. The significance of such complexity is now beginning to understand. We started to understand what is happening, why the complexity is happening. It's all because of the different types of the enzymes we have, that is RNA polymerases 1, 2, 3, and the process of processing of hnRNA into mRNA. And that is what we said already, split gene arrangement. The split gene arrangement is a reason for which it has to undergo processing, splicing, where removal of introns takes place and addition of exons takes place. These are exons which has to be added together, right? Now, the split gene arrangement represents probably an ancient feature of the genome. While we are discussing about the RNA world, we already said that the RNA begins as its genetic material, then it is transferred, modified into DNA because the DNA structure is stable, more stable when compared to the structure of the RNA. That is what the nucleic acid DNA has been selected as a genetic material which stores the genetic information, which stores the hereditary information. The presence of the introns is a reminiscent of antiquity. Ancient. It is a remembrance of the ancient that RNA is acting as a genetic material initially, then the genetic material is further transferred to be in DNA. And the process of splicing represents the dominance of RNA. It is the RNA nucleic acid which is acting as a genetic material initially. Then modifications happens towards the increasing of the stability of the genetic material, and finally it is DNA. In recent times, the understanding of RNA and RNA dependent processes in the living system has assumed more importance. All the research is towards to understand more about RNA now. We have understood enough about the DNA. We know that DNA is a more stable structure, and it is a very apt nucleic acids to be act as a storage of the genetic information. Now the research is also going towards understanding of the RNA. That is what we consider in an RNA world, right? So this is the representation where it is the process of transcription that is happening in the eukaryotic cells. In the eukaryotic cells, major difference is the clear-cut division of the labor, where we see different types of enzymes like RNA polymerases 1, 2, 3. So here, similarly, one of the strand is going to act as a template, and that is what, using that template, and the rule here is complementarity. Once the promoter reaches to the RNA polymerases, that is to the promoter region, RNA polymerases go and fix and initiate the process of transcription. Once the transcription initiates, it leads to the elongation of the transcription, and it terminates, which leads to the formation of mRNA. But here this mRNA is primary transcript, what we call it as an hnRNA. In hnRNA, heterogeneous nuclear RNA, we do the presence of exons. These are exons. These are exons. These are all exons. And the other side, intron, introns, 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 introns. These all introns are has to be removed. These introns will remove so this is the intron now. It will be removed. See, these are all the introns which are removed from the hnRNA. Meanwhile, capping as well as tailing is happening. What do you mean by capping? Towards the 5' end, methyl guanosine triphosphate is getting added. And towards the 3' end, poly A, that is many adenylates, polyadenylate units, residues of approximately 200 to 300 adenylate units will get attached to the 3' end. So towards the one end we have methyl guanosine triphosphate and the other end polyadenylate. So this is a tail and this is capping. We have seen splicing, we have seen capping, we have seen polyadenylation, that is adding up of polyadenylate residues. So once all this happens, now we call it as a process mRNA. So from here, DNA, the genetic material has been copied into the mRNA, and all this process is called as transcription that is happened in the eukaryotic cell. But when compared to the prokaryotic cell, where in the prokaryotic cell we don't have any differentiation of the compartmentation, both the process takes place within single, that is cytosol of the cell, and there is no splicing, there is no tailing, there is no capping. That is it. So today we understood about the different types of the RNAs that are useful for the protein synthesis, that is rRNA, tRNA, mRNA. And also we discussed about the process of the transcription happening in prokaryotic cells as well as eukaryotic cells. Right? With this, we come to the end of the session. Let's meet up in the next coming up session. Until then, keep learning with We Learning. Thank you.